Hello and welcome everyone to this podcast where we're going to continue our discussion of experiments that show that DNA was the genetic material in organisms that contain DNA. We're going to begin by talking about Hershey and Chase. I want to begin by talking about experimental techniques that we could use, or that they could use in the 1950s when these experiments were done, to measure the amount of protein or the amount of DNA in a particular sample. At this time, radioactive isotopes were becoming increasingly more accessible and usable in a laboratory situation. And so if you wanted to measure proteins and distinguish them from DNA, there had to be something in the structure of a protein that was unique to it that DNA didn't have, and something in the structure of DNA that DNA had but proteins did not have. And that structure for proteins is sulfur. And the radioactive isotope for sulfur is S35. And there's no sulfur found in DNA. Sulfur is a good one to use because sulfur is found in proteins because of amino acids like methionine and cysteine, which both contain sulfur. Sulfur is never found in DNA. For DNA, they use radioactive phosphorus, and its radioactive isotope is P32. Phosphorus is a good choice because phosphorus is found in DNA. As we talked about the structure of DNA, it always has phosphorus at those phosphodiester bonds, and phosphorus is not typically found in proteins. So what this means is if I have a flask or beaker here containing P32, and I have a flask with S35 in it. Now let's draw a third container, and this container has no radioactive phosphorus or sulfur in it. I can easily distinguish which ones have radioactivity in it by using a Geiger counter. A Geiger counter is a tool used to measure radioactivity. If it's close to a substance or material that has radioactivity in it, it's going to make a dinging noise repeatedly over and over again, and the closer you get to it or the more that's present, it's going to beep louder and with shorter time in between each of the beeps. So we took this Geiger counter and we held it over the radioactive phosphorus here. We would see it make a bunch of dings. If I held it over the S35, I would also see many dings coming from it. If I held it over our control with no radioactivity in it, I would see nothing, or I would hear nothing. Now the Geiger counter isn't going to be able to distinguish between P32 and S35, but it'll tell you if one or the other is present. And so they use this technique in their experiments. Now there's one more thing we need to talk about before we talk about their experiment, and that is the organism that they worked with. And the organism they worked with is called Bacteriophage. We'll talk about this organism a little bit later in more detail when we talk about the genetics of viruses. But it's important to talk a little bit about it now because it's the organism they used. And a bacteriophage is a virus that attacks bacteria. So let's draw a bacterial cell here. And the bacteriophage will attach to the bacterium, the E. coli cell, and it will release its genetic material into the bacterium. Inside the bacterium, the bacteriophage will assemble new viruses. using the E. coli's protein synthesis machinery. We now know that this is DNA, but at the time they did this experiment, they didn't know it was DNA. So it would take the DNA and make new bacteriophage, and a lot of them. So it would make a lot of these, until at some point it's full of these bacteriophage, and then, for lack of a better way for me to draw it, the E. coli bursts and releases all of these bacteriophage. And now they can go on to infect other bacteria. So this seemed like a very simple organism for Hershey and Chase to use to show that DNA was the genetic material in these bacteriophage. Because they knew that in a bacteriophage, and these bacteriophage right here, they were about 50% DNA and 50% protein. So if they could find some way to label the DNA and some way to label the protein, then they could analyze their results. And so that's what they did. They were able to label the proteins with S35 and the DNA with P32. So now let's talk about the experiment and how they did it and how we can interpret the results. Now, using these bacteriophage and the knowledge of the radioactive sulfur and phosphorus, they could conduct their experiment. Let's first talk about the experiments they used with radioactive sulfur, S35, and remember that labeled proteins. They obtained bacteriophage that contained this radioactive sulfur in it, meaning all the protein in this bacteriophage were radioactive. None of the DNA was radioactive. So if you held a Geiger counter over these bacteriophage, it would make the clicking noise indicating it was radioactive, and since we only put S35 in it, we know it means that the proteins are radioactive and not the DNA. They then took the bacteria, the E. coli cells, and allowed it to infect the bacteria. It would inject its genetic material into the E. coli. So I'm just going to draw a line here for the genetic material and just write GM. At this time, they didn't know if it was protein or DNA. But if it was protein, then the radioactive protein would have went in there. If it was DNA, then the radioactive protein would have stayed in the phage, and the non-radioactive DNA would have went into the cell. They allowed them to grow for a specific amount of time that they knew was the exact amount of time necessary for the genetic material to get inside the cell. They then took a blender, just like a regular blender you might have in your kitchen to make milkshakes with. That's my blender. And they put these in here, the whole culture in there, not just one of course, the whole culture, and they turned it on. And in turning it on, what that would do is it would remove the bacteriophage from the E. coli, because they're not attached there by strong covalent interactions, they're just precociously attached, and so they're going to fall off pretty easily. So they spun it really quick, and then they dumped this off into centrifuge tubes, and I'm going to draw these centrifuge tubes fairly large. They're not really this big, but I need to be able to draw inside of it. Inside of this centrifuge, the stuff that was left from the blender, inside the centrifuge tube, there would be E. coli cells, lots of them, right? And inside of them would be the genetic material. DNA or protein, they didn't know, but the genetic material would be inside of them. And the empty phage particles would have been removed. And they call these empty phage particles ghosts. They no longer have genetic material, they can no longer infect anything, so they're called ghosts. They knew if they took this centrifuge tube and spun it down at a certain speed, what they would end up with, so let's go centrifuge here. They would get their centrifuge tube here. At the very bottom of it would be the cell pellet. And this contained E. coli, the bacteria, plus the phage, the bacteriophage genetic material, because it's inside of the E. coli. What would be in the supernate, this part here, the liquid part, that didn't form in the pellet, would be the ghost bacteriophage, all of them. So now they could take this supernate here, so let's write over here, supernate, and they could wave the Geiger counter over it, and if it beeped, then they knew the protein had to be in here. They could take the pellet, which contained the genetic material, and wave the Geiger counter over it, and if it beeped, then they knew that the protein was in the E. coli and likely the genetic material. So let's make a quick little table here. Radioactivity. So when they waved the Geiger counter over the supernatant here, they saw radioactivity, which meant that the protein was still in these ghosts. Because remember, they used S35. S35 only labels proteins. So if the radioactivity was here, it said that the it said that the proteins had to still be in the coat, in these ghosts. Now when they put the Geiger counter over the pellet, what they discovered was no radioactivity, meaning no protein. Rather, no phage protein in this pellet or in the E. coli. These experiments suggested that protein is not the genetic material. I ran out of room, but material. Now, you can't just assume DNA has to be the genetic material at this point. You have to do the other experiment. We have to look at P35. So I'm not going to erase all of this. I'm just going to erase the parts I need to erase to do the P32 experiment. So let's erase this, and we'll erase this. Okay, so the experiment's done the exact same way. They take radioactive phosphorus, P32, and they grow bacteriophage in its presence so that this bacterium has, this bacteriophage has P32 in it. Its DNA is labeled. Its protein is not. So let's write that here. DNA is radioactive, but the protein is not. So when we're doing these experiments, we're only looking at radioactive DNA. Protein's still there, it's just not radioactive. Again, they allow the bacteriophage to infect the E. coli cells. They wait a specific amount of time. They flip on the blender, which shears off the bacteriophage, creating these ghosts that no longer contain the genetic material, and the E. coli that now contain the genetic material of the bacteriophage. They then centrifuge it. In the pellet is going to still be the E. coli plus the phage genetic material. In the supernate is going to be the ghost phage, lacking the genetic material because it put all the genetic material into the E. coli. Now, using radioactive phosphorus, when they take their Geiger counter and they wave it over the supernate, they see no radioactivity, so no radioactive DNA. So no radioactive DNA. But when they wave it over the E. coli cells, the Geiger counter over the E. coli cells, they do see radioactive DNA. So from this experiment, we can conclude that not only is protein not the genetic material for the bacteriophage, but that DNA is the genetic material. So between Avery, McLeod, and McCarty's experiments with the Streptococcus bacteria infecting the mice, and the experiments here by Hershey and Chase with using bacteriophage that infect E. coli cells, the scientific community was willing to accept that DNA was the genetic material. There are also many other experiments that were done that also supported these claims as well. And essentially that's the process of science. Over time, more and more experiments are done, which leads to more evidence and eventually accepting of the hypothesis. Now the question that kept nagging people is that there are some organisms that lack DNA. So if they lack DNA, yet they still behave as if they have a genetic material, then what would the genetic material be in those organisms? And this is where Geiger and Schramm come in, because they worked with an organism called tobacco Mosaic virus. So viruses aren't true cells, and they're not truly living, but they possess the ability to reproduce and the ability to infect other cells. Unlike the bacteriophage, though, the tobacco mosaic virus, which we'll just refer to TMV, it doesn't have any DNA. It's a very simple structure. It contains an RNA molecule, no DNA, an RNA molecule, surrounded by a protein coat. So this virus now would infect a tobacco plant. Now obviously these are not proportional sizes, the virus is much, much smaller than the leaf. But it would infect these tobacco leaves, and on the tobacco leaves it would create more viral particles and eventually destroying the plant leaves, allowing for more viruses to be produced. So in these organisms, what's the genetic material? So which is the genetic It has to be RNA or protein because that's all that's present. So what is it? So what Geiger and Schramm did was they would take the intact virus with the RNA in it, and they would put that on a tobacco leaf. And what they would get, and this was known before, they would get more viruses growing on the leaf. So the next thing they did was they would just take the protein. They would remove the RNA and they would just have the protein of the virus. And they would put that on the tobacco leaf. And what they got in return was no new viruses. Next, they took the RNA from the tobacco mosaic virus without the protein, just the RNA, and they put that on the tobacco plant. And what they got was more viruses. It was able to, with just the RNA, it was able to produce more TMV viruses. From this, they concluded in TMV, RNA is the genetic material. It could also have been suggested, though not proven at this time, that all RNA viruses use RNA as their genetic material. It has subsequently been shown to be true. So this is a very important experiment showing that RNA could act as the genetic material, and in fact RNA probably was the first genetic material before DNA was around. In class, we're going to talk about some indirect evidence that supported that DNA was the genetic material in organisms like humans, because in humans we can't quite do the same experiments that we would do with the bacteria and with mice. So that ends this podcast, but just a quick summary. We talked about the Hershey and Chase experiments, where they were able to conclude that DNA is the genetic material of bacteriophage. And equally important, that protein was not the genetic material of bacteriophage. And these experiments, for the most part, put it to rest that DNA was the genetic material. Next, we talked about Geyer and Schramm. And Geyer and Schramm showed that in organisms, in viruses I should say, that are RNA viruses, they lack DNA. So in these RNA viruses, they showed that RNA was the genetic material in the TMV, tobacco mosaic viruses, and probably really all RNA viruses, but they didn't test them all. Your book describes some other experiments that they used with these RNA viruses, but this is the only one we've talked about. As you go forward trying to study for this material, I think it's a useful exercise to ask what the results of the experiments would have been if proteins were the genetic material, or if RNA was the genetic material, or if carbohydrates or lipids were the genetic materials. Could these experiments have been done in such a way that would have allowed us to show that those macromolecules were the genetic material? Or if we isolated a sample from another planet that had life, how would we show that DNA was the genetic material in those newly discovered organisms? Alright, that's all I have for this podcast. If you have any questions, please let me know, and I'll help answer any questions you might have. Have a good one, and see you in class.